Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover some of the most extreme marine creatures that live in the oceans. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. The Chrysomalin squamiferum, the volcano snail, lives 2,900 meters deep next to black smokers that shoot out water at 350 degrees Celsius, and it's the only animal that builds its own skeleton out of iron sulfide. It's the first on a list of organisms that made biologically possible what should be impossible. The Pompeii worm, Alvinella pompejana*, with a 78 degrees Celsius temperature difference between its head and tail at the same time. The Yeti crab, Kiwa hirsuta*, which farms bacteria on its own arms like a portable garden. The giant tube worm, Riftia pachyptila*, 3 meters long with no digestive organs whatsoever. The ice fish, Kinocephalus aceratus*, the only vertebrate without hemoglobin with colorless blood and a heart 5 times larger than that of comparable fish. The Dumbo octopus, Grimpeteuthis*, spp, which floats at 7,000 meters with ear-shaped fins. The transparent sea cucumber, Gnypnias saximia*, which breathes through its anus and sheds its own luminescent skin when touched. The vent mussel, Bathymodylis*, spp, the only bivalve with simultaneous dual chemosynthetic symbiosis. And the Greenland shark, Somniosus microcephalus*, which takes 150 years to reach maturity and carries parasites that destroy its vision while it swims at 3 kilometers per hour in polar darkness. The volcano snail's shell has 3 layers. The outermost is made of iron sulfides, the middle one is organic, and the innermost is aragonite, a materials engineering feat that no human technology has been able to replicate. Picture finding, at the bottom of the ocean, an animal wearing iron armor forged by the planet itself. Not a metaphor, actual mineral, deposited layer by layer around a soft, vulnerable body. Chrysomalin squamiferum, formally described in 2001 on the central ridge of the Indian Ocean, lives near black smokers. Cracks in the ocean floor that shoot out superheated fluids loaded with dissolved minerals. When those fluids hit the cold seawater, metallic compounds precipitate and pile up around the opening, building chimney-shaped structures that can reach dozens of meters tall. There, where pressure equals hundreds of atmospheres and temperature swings between deadly extremes, this remarkable gastropod thrives with a biochemistry that has no known parallel in the animal kingdom. The outer shell of the volcano snail is made of gragite and pyrite, iron sulfides that biology doesn't normally use to build support structures. Other animals use calcium carbonate, calcium phosphate, or chitin. None besides this species systematically incorporates iron sulfide into its exoskeleton. These minerals reach the organism through chemosynthetic bacteria, living in a specialized gland in the animal's esophagus. These bacteria metabolize sulfur compounds found in hydrothermal fluids and supply, as a byproduct, the sulfur and iron needed for biomineralization of the outer layer. The animal doesn't just live in a volcanic environment, it structurally incorporates it. The middle layer of the shell is organic, made up mainly of conchiolin, a fibrous protein that acts as a shock absorber between the outer mineral armor and the inner aragonite layer. This 3-layer architecture is no accident. Mechanical tests carried out by MIT researchers showed that the specific arrangement of these layers distributes and dissipates impacts better than synthetic materials designed for equivalent purposes. When the iron sulfide layer takes a hit, The energy spreads laterally through the organic layer before reaching the aragonite. The damage gets fragmented and absorbed instead of concentrated at a single breaking point. No industrially manufactured composite material has replicated this mechanism to an equivalent degree to this day. Scales covering the animal's foot, the muscular structure the gastropod uses for movement, are also mineralized with gragite. This scaly coating is entirely unique among mollusks. To picture what that means, imagine wearing shoes made of volcanic rock that, at the same time, protect, flex, and allow movement. The volcano snail's scales articulate in a similar way, providing protection without sacrificing mobility. The animal's characteristic black coloration comes from the gragite. Specimens raised experimentally without access to sulfur compounds develop translucent scales, confirming the mineral origin of the pigmentation. The black smokers surrounding the volcano snail's habitat shoot out fluids at temperatures reaching 400 degrees Celsius, above the boiling point of water at sea level, but the abyssal pressure keeps the fluid in liquid form. As it mixes with the surrounding seawater, which is close to 2 degrees Celsius, it creates violent thermal gradients that concentrate minerals and sustain entire biological communities based on chemosynthesis, the process by which organisms produce energy from chemical reactions instead of sunlight. The volcano snail lives right in that mixing zone, where temperature and chemical composition shift abruptly over just a few centimeters. Its tolerance for these extreme gradients depends on molecular adaptations that are still only partially understood. Proteins involved in the volcano snail's biomineralization process are being studied as models for developing new engineering materials. The biological ability to deposit minerals with nanometric precision in hierarchical structures that optimize mechanical strength while maintaining flexibility surpasses the capabilities of current industrial processes. Conventional materials face a constant trade-off. Greater hardness generally means greater brittleness. This animal's shell resolves that dilemma through functional layering. Each layer plays a distinct role, and the combination produces emergent properties that no individual layer would have on its own. The species was listed as vulnerable by the International Union for Conservation of Nature in 2019, making it the first animal threatened by deep-sea mining before any operations had even begun. The hydrothermal fields it inhabits contain significant concentrations of rare metals, cobalt, nickel, copper, that are of interest to the deep-sea mining industry. 3 of the 4 known populations are located in areas with active mineral exploration licenses. The irony is twofold. The animal that incorporates minerals like no other faces an existential threat precisely because of the minerals around it. Understanding how the volcano snail regulates iron deposition requires understanding its symbiosis with esophageal bacteria at the molecular level. These bacteria, belonging to the phylum Gammaproteobacteria, oxidize hydrogen sulfide for energy, producing organic compounds that feed the host and indirectly supply precursors for mineralization. The animal doesn't actively direct this process. The biochemical architecture was shaped over generations by intense selective pressure in an environment where any failure in protection means immediate death by predation or being crushed by hydrothermal fluids. The pressure at 2,900 meters is roughly equivalent to 290 atmospheres. Every square centimeter of body surface bears a force equal to the weight of a compact car. Abyssal organisms generally compensate by adjusting the composition of cell membranes, increasing fluidity to maintain function under conditions where conventional membranes would solidify. Proteins fold in slightly different ways. Enzymes have active sites calibrated for pressures that would destroy their surface-level equivalents. The volcano snail stacks all of these deep molecular adaptations while, at the same time, keeping active minerosynthesis running in one of the most chemically aggressive zones on the planet. All of the volcano snail's biology converges on one unsettling conclusion. What we call inhospitable conditions is just a matter of perspective. For this species, black smokers aren't a catastrophe, they're infrastructure. The iron that would poison surface organisms becomes armor. The sulfur that would choke lungs feeds the bacteria that keep the animal alive. Every element of the lethal environment has been biologically repurposed into a resource. And while materials engineers search their labs for solutions that nature already worked out in black shells 3 kilometers down, the volcano snail keeps doing what it's always done, existing where nothing should, protected by armor the earth itself forged. Alvanella pompejana builds tubes directly on the walls of black smoker chimneys in the eastern Pacific, 2,500 meters down. Specimens have been found with one end of their body in water 78 degrees Celsius hotter than the other end, the steepest temperature gradient tolerated by any animal on the planet. The head, with its scarlet gills, sits exposed to water around 22 degrees Celsius, while the tail sits in water close to 80 degrees Celsius. The confirmed upper thermal limit in lab settings is below 55 degrees Celsius, but the thermal optimum is above 42 degrees Celsius, confirming that the Pompeii worm is one of the most heat-tolerant metazoans ever documented. A dense mat of bacteria covers the worm's back, possibly functioning as biological insulation against extreme heat, a mechanism that hasn't been fully confirmed yet. Picture splitting your body in two, half submerged in near-boiling water, half cooled to pool temperature. Not for a few seconds, permanently, for your entire life. That's the everyday reality of Alvanella pompejana, a polychaete discovered in 1980 during explorations of the Galapagos Ridge in the eastern Pacific. The name references Pompeii, the Roman city buried by a volcanic eruption, because the worm's environment, blanketed in mineral deposits and permeated by extreme heat, inevitably evokes that catastrophe. The difference is that Pompeii killed. The Alvanella's environment is its functional home, fine-tuned by biology over geological time to withstand conditions that would destroy any conventional animal tissue in seconds. The Pompeii worm's body measures between 10 and 13 centimeters long. That seems modest for an organism that holds animal heat tolerance records. It lives in tubes made of material secreted by specialized glands, fixed directly to the sulfide chimney walls that black smokers build up. These tubes provide partial insulation, but the internal temperature still swings dramatically between the front opening, where the head with its scarlet gills stays exposed to relatively cold seawater, and the back end, in direct contact with the chimney wall heated by hydrothermal fluids. This housing architecture creates the most extreme thermal gradient ever documented in any animal. Proteins are the biological structures most vulnerable to heat. Enzymes, antibodies, cell receptors. All of them depend on precise three-dimensional shape to function. High temperature breaks the weak interactions that hold that shape together. The protein unfolds, loses function, precipitates. This process is called denaturation, the same phenomenon that turns liquid egg white into a solid white mass when you cook it. Organisms adapted to extreme heat, called thermophiles, have proteins with extra bonds that resist denaturation at higher temperatures. Alvanella pompejana needs functional proteins in tissues that simultaneously experience 22 degrees Celsius and 80 degrees Celsius, a biochemical challenge with no equivalent in the animal kingdom. The bacterial mat covering the Pompeii worm's back is one of the most studied and still least understood symbiotic systems in abyssal biology. Long filaments of epsilon proteobacteria form a dense layer over the animal's dorsal surface, visible to the naked eye as a white, fuzzy texture. The leading hypothesis suggests this layer acts as thermal insulation, slowing the rate of heat transfer between the hot external environment and the host's internal tissues. An alternative proposal is that the bacteria metabolize toxic compounds from the hydrothermal fluid before they can penetrate the tissues. Both mechanisms may be operating at the same time. Definitive experiments are made difficult by the impossibility of keeping the organism alive in a lab for any extended period. Capturing and studying Alvinella pompigana under controlled conditions is notoriously hard. Bringing the animal to the surface means rapidly dropping it from 290 atmospheres to normal atmospheric pressure, a process that expands dissolved gases in tissues, causes cellular swelling, and destroys internal structures. Pressure-controlled systems exist, but maintaining the specific thermal gradient while carrying out precision biochemistry adds considerable layers of technical complexity. Much of what's known about the Pompeii worm's physiology has been inferred from specimens collected under suboptimal conditions or from genomic analyses performed on material preserved immediately after capture. The Alvinella pompigana genome has been partially sequenced and reveals expansions in gene families associated with heat stress response, particularly genes encoding heat shock proteins, HSPs. These proteins function as molecular chaperones. They bind to partially denatured proteins and help them refold correctly before the denaturation becomes irreversible. Organisms in stressful environments typically express HSPs at high levels as a continuous protection mechanism. In the Pompeii worm, the expression of these proteins appears to be constitutive, permanently active rather than only triggered in response to temperature spikes. It's like keeping firefighters inside the building on full-time shifts instead of calling them only when the alarm goes off. The scarlet gills of Alvinella pompigana owe their color to hemoglobin, the oxygen-carrying protein that is red in most vertebrates but takes on varied functions and colors in polychaetes depending on the species. These gills stay exposed to the cooler water, where dissolved oxygen is more abundant. The worm's body transports that oxygen from the cold head to the posterior tissues exposed to the heat, maintaining aerobic metabolism in cells operating near extreme thermodynamic limits. Any failure in that transport, circulatory blockage, local hemoglobin denaturation, can be rapidly fatal in an environment where the biological margin for error is razor-thin. The Alvinella pompigana's environment is also chemically hostile. Hydrothermal fluids carry hydrogen sulfide at concentrations that are directly toxic to most animals, inhibiting the enzyme cytochrome C oxidase, an essential component of cellular respiration. The worm neutralizes part of that problem through symbiotic bacteria that oxidize sulfide as an energy source, reducing the concentration available to penetrate its tissues. Heavy metals, copper, zinc, lead, also concentrate in the fluids, requiring detoxification mechanisms that the animal keeps running continuously. Every chemical challenge is managed by parallel systems, forming a network of protections that all operate at the same time. The Pompeii worm exists as a hands-on demonstration of the limits of biological adaptation, and of just how arbitrary those limits seem when you confront actual living organisms. All of animal physiology was described, for decades, as operating within relatively narrow thermal ranges. Alvinella pompigana inhabits a range that crosses multiple categories at once. Its head and its tail could, thermally speaking, belong to two animals from completely different environments. And yet the organism integrates those conditions into a coherent, functional, reproducing system. What seems impossible isn't impossible. It's just rare enough that biology needed volcanic fissures 2,500 meters deep to prove it. Kiwa hirsuta was discovered in 2005 off Easter Island, 2,200 meters deep, and was so unlike any known crustacean that it established an entirely new family. Its arms are covered in long, dense bristles, the setae. The Yeti crab cultivates chemosynthetic bacteria in those bristles, rhythmically waving its arms near hydrothermal vents to maximize bacterial growth, and then eats them. The Antarctic species Kiwa tyleri forms aggregations with up to 4,017 individuals per square meter, a density comparable to social insect colonies, in an environment where any drift away from the heat source means immediate death from polar cold. Discovering an animal different enough to justify an entirely new taxonomic family is a rare scientific event. Families are broad categories. The family Felidae includes everything from house cats to lions. The family Hominidae includes humans and chimpanzees. Creating a new family means recognizing that an organism has diverged from everything else so fundamentally that no existing category adequately captures its biology. That's what happened with Kiwa hirsuta in 2005. The family Kuwaiti was established specifically for this crustacean, whose arms covered in bacterial bristles represented not just unusual morphology, but a feeding strategy with no documented precedent among arthropods. The setae, bristles covering the chelipeds, modified arms, of the Yeti crab, are rigid filamentous structures made up primarily of chitin, the same polysaccharide that forms the exoskeleton of all arthropods. In other crustaceans, setae typically function as feeding filters or sensory organs. In Kiwa hirsuta, each individual seta serves as an attachment substrate for chemosynthetic bacteria that colonize the external surface of the filament. The density of this colonization is enough to make the arms visibly furry, a feature that inspired the popular name Yeti crab, after the supposed furry hominid of Himalayan folklore. Chemosynthesis is the process by which certain bacteria produce organic energy from inorganic chemical reactions, without depending on sunlight. In hydrothermal environments, bacteria oxidize hydrogen sulfide or methane, compounds abundant in the fluids that emerge through cracks in the ocean floor, obtaining energy to fix carbon and build biomass. This bacterial biomass forms the base of the entire hydrothermal food chain. Just as land plants capture solar energy and pass it on to herbivores, chemosynthetic bacteria capture chemical energy and pass it on to larger organisms. The Yeti crab cut out the middleman by farming those bacteria directly on its own body. The arm-waving behavior, documented in footage captured by remotely operated vehicles, is no random movement. Hydrodynamic flow analyses suggest that the rhythmic motion of the chelipeds maximizes the exposure of the colonizing bacteria to the sulfide-rich fluids emerging from the hydrothermal vents. The bacteria need these compounds to carry out chemosynthesis. Depriving the setae of hydrothermal fluid would reduce bacterial growth rates and, consequently, the availability of food for the host. The Yeti crab, therefore, doesn't just carry a bacterial garden. It actively irrigates it with nutrients by positioning and moving its arms in a way that appears optimized for microbial productivity. The species Kiwa tyleri, described in 2012 from specimens collected at the South Georgia Islands in Antarctica, shows behavioral variation that amplifies the genus's peculiarity. In this species, thermal constraints are even more severe. Antarctic hydrothermal vents exist in surrounding water close to 0 degrees, and drifting away from the vent opening means immediate exposure to lethal cold. This forces individuals to crowd into extremely narrow bands around the heat sources, generating record population densities of more than 4,000 individuals per square meter. Crustaceans that are normally solitary become, out of thermal necessity, practically gregarious. The Yeti crab's diet supplements bacteriophagy, consumption of the bacteria farmed on its arms, with opportunistic predation and possibly scavenging of carcasses available in the environment. Isotopic analyses of Kiwa hirsuta tissues revealed chemical signatures consistent with consumption of both chemosynthetic bacteria and organic material from other sources. This dietary generalism is an advantage in an environment where resource availability can shift abruptly as vent activity fluctuates. The bacterial farm on the arms functions as a portable food reserve. Additional sources supplement the diet when available and necessary. The white exoskeleton and the absence of functional eyes. Kiwa hirsuta has vestigial, unpigmented eyes, structurally present but functionally rudimentary, reflect adaptations common to abyssal organisms. Without sunlight penetrating to 2,200 meters, protective pigmentation against ultraviolet radiation is unnecessary and energetically costly. Sophisticated eyes, likewise, offer minimal adaptive return in permanent darkness. Selection tends to reduce structures that consume resources without providing any advantage. Over generations, functional eyes regress and pigmentation disappears. The Yeti crab navigates its environment through mechanoreceptors in its antennae and in the setae themselves, sensitivity to water flow, fluid chemistry, temperature. The discovery of Kiwa hirsuta reignited discussions about the limits of feeding strategy diversity in animals. Agriculture, the deliberate cultivation of other organisms as a food source, was considered uniquely human until detailed observations of leafcutter ants farming fungi, termites maintaining fungal gardens, and ambrosia beetles transporting spores. The Yeti crab adds a marine, abyssal, chemosynthetic case to that list. While intentionality in the cognitive sense can't be attributed to the animal, the arm-waving behavior constitutes, functionally, a form of active bacterial culture management, agriculture by any objective ecological standard. The Yeti crab closes the loop that the volcano snail opens. Both inhabit hydrothermal vents. Both depend on bacterial symbioses to survive where the conventional food chain doesn't reach. Both incorporate their environment into their biology in ways that challenge previously established categories. Between them, the Pompeii worm endures gradients that should shred proteins. The Yeti crab turns its own limbs into farms. Each organism, on its own, would be enough to redefine what we understand as a biological limit. Together, they make an unbroken argument. The conditions that seem impossible are, more often than not, simply the conditions where life hasn't yet been looked for carefully enough. Riftia pachyptila was discovered in 1977 at the Galapagos hydrothermal vents and immediately challenged everything biology knew about animal nutrition. It has no mouth, no intestine, and no anus, relying exclusively on internal bacteria for nourishment. Those bacteria make up more than half of the animal's total body weight. The red plume at the top of the tube works like a gill. It absorbs hydrogen sulfide, CO2, and oxygen from the water and delivers them to the internal bacteria, which convert these compounds into energy through chemosynthesis. It's the fastest growing marine invertebrate. It can grow more than 85 centimeters in a single year. An animal 2 meters long with no mouth and no stomach, fed by billions of bacteria inside an organ called the trophosome. Every animal studied before 1977 ate in some way. Mouth, tentacles, filters, absorption of dissolved organic matter. Nutrition was, by definition, a process of ingestion. The discovery of Riftia pachyptila at the Galapagos hydrothermal vents forced a revision of that axiom. Here was an animal over 1 meter long, eventually reaching 3 meters, the largest known tube-dwelling marine invertebrate, with no food intake opening, no digestive cavity, no waste outlet. Biology had found an organism that had completely outsourced digestion to internal tenants, making the digestive tract evolutionarily redundant to the point of total anatomical disappearance. The tube surrounding the Riftia pachyptila body is built by the animal itself from chitin and secreted proteins, forming a rigid white structure that can reach 5 centimeters in diameter. This tube is no passive housing. It's a functional component that anchors the animal to the rocky substrate near hydrothermal vents and protects soft tissues from abrasion and predation. The animal can fully retract inside the tube when it detects a disturbance. The red plume that emerges from the upper opening, a structure called a tentacle or gill crown, is the only part of the animal regularly exposed to the external environment during normal feeding. The intense red color of the plume comes from hemoglobin, but not the hemoglobin familiar from vertebrates. Riftia pachyptila has extracellular hemoglobins with an extraordinarily complex molecular structure, circulating freely in the vascular fluid rather than confined to cells. These molecules have a unique property. They bind oxygen and hydrogen sulfide simultaneously without the two compounds interfering with each other. In virtually every other organism, hydrogen sulfide is a metabolic poison that inhibits the enzyme cytochrome C oxidase, blocking cellular respiration. Riftia's specialized hemoglobin sequesters the sulfide, safely transports it through the body, and delivers it to the bacteria without poisoning the surrounding tissues. The trophosome, an organ that takes up much of the animal's internal volume, has no equivalent in any other animal phylum. It's a highly vascularized tissue that houses between 285 and 385 million bacteria per cubic millimeter of tissue. These bacteria, belonging to the genus Candidatus Endoryptia Persephone, carry out chemosynthesis. They oxidize hydrogen sulfide for energy and use that energy to fix carbon dioxide into organic compounds, which they then transfer to the host. The trophosome is functionally an internalized gut where digestion has been replaced by bacterial primary production. The plant and the herbivore fused into a single organ. Riftia pachyptila's growth rate defies intuitions about abyssal metabolism. Deep ocean floor environments are generally characterized by slow growth. Scarce resources, low temperatures, and high pressures impose severe metabolic constraints. Riftia grows more than 85 centimeters in a single year, making it the fastest-growing marine invertebrate on record. That speed reflects the efficiency of bacterial chemosynthesis as an energy source. Hydrothermal vents provide a continuous and abundant flow of hydrogen sulfide, eliminating the scarcity that typically limits growth in abyssal environments. The animal doesn't depend on the rain of organic matter sinking from the surface. It has its own internal power plant. Communities of Riftia pachyptila form dense aggregations around active hydrothermal vents, with interlocking tubes creating three-dimensional structures that other organisms inhabit. Crabs, fish, polychaetes, and a variety of mollusks colonize the space between the tubes, depending directly or indirectly on the bacterial productivity the worms sustain. When a hydrothermal vent goes extinct, a process that happens on timescales of decades to centuries, that entire community collapses. Riftia is a foundation species. Without it, the structured habitat that supports dozens of other species simply doesn't exist. Its singular biology carries ecological consequences that ripple through the entire local food web. The transmission of symbiotic bacteria between generations of Riftia pachyptila occurs horizontally. Larvae are born without bacteria and acquire them from the environment during their juvenile phase. Newly hatched larvae have a temporary functional digestive opening, used to capture free-living bacteria present in the water near hydrothermal vents. Once the symbiosis is established, that opening closes permanently and the digestive tract regresses through development. The animal literally rebuilds its internal anatomy after acquiring its symbionts. The presence of the bacteria triggers trophosome differentiation and the regression of digestive organs, as if the genetic instruction was waiting for confirmation of the partnership before committing to the final body plan. Pressure at 2,500 meters, roughly 250 atmospheres, poses no obstacle to Riftia pachyptila because its tissues, like those of all adapted abyssal organisms, contain no compressible gas cavities. Lungs, swim bladders, and air-filled cavities collapse under intense pressure. Solid tissues, or those filled with incompressible fluid, transmit pressure uniformly without structural deformation. Riftia resolves this by being essentially solid. The dense trophosome, fluid-filled vascular vessels, and musculature distribute pressure without concentrating it at breaking points. The same physical principle that protects deep-sea organisms in general applies here with an added wrinkle. The animal must maintain precise internal chemical gradients under pressure that, in less integrated systems, would homogenize everything. Riftia pachyptila started out as a baffling anomaly and became a model organism for studying the origins of obligate symbioses, the evolution of body plans, and the possibility of life in chemosynthetic environments beyond Earth. Moons like Europa and Enceladus have subsurface oceans potentially rich in reduced chemical compounds, exactly the kind of environment where chemosynthesis is viable. If life exists on those moons, it may have converged on strategies similar to Riftia's. Organisms that extract energy from geochemical reactions without depending on sunlight. The mouthless worm of the Eastern Pacific became, inadvertently, a biological argument for the search for life on icy worlds of the solar system. The icefish, Kinocephalus aceratus*, lives in the polar waters of Antarctica and is the only adult vertebrate without hemoglobin, the protein that transports oxygen in the blood of virtually every animal with a backbone. To compensate for the absence of hemoglobin, icefish have larger blood vessels and low-viscosity blood volumes 4 times greater, and hearts with cardiac output 5 times higher compared to other fish. These fish have translucent blood, white hearts, and have adapted to live without red blood cells. Oxygen dissolves directly into the plasma, a strategy that only works because the extremely cold Antarctic water retains more dissolved oxygen. Removing this fish from the cold environment is enough to kill it from lack of oxygen in the plasma. Hemoglobin is one of the oldest and most conserved proteins in animal biology. Present in worms, mollusks, insects, and virtually all vertebrates, this iron-based molecule evolved as a universal solution to a universal problem, transporting oxygen from where it's absorbed to where it's consumed. Without hemoglobin, blood plasma dissolves oxygen directly, but in negligible amounts, insufficient to sustain active metabolism. Every known vertebrate carries hemoglobin in dedicated red blood cells, except one. The family Channicthiidae, made up of 16 species of Antarctic icefish, functionally lost its hemoglobin genes over millions of years and survived by completely rebuilding its cardiovascular physiology to compensate for an absence that should have been lethal. The water of the Antarctic Ocean near 0 degrees dissolves roughly twice as much oxygen as tropical waters at 25 degrees Celsius. This physical fact, described by Henry's law, which relates gas solubility to temperature, is the foundation that makes the existence of icefish possible. Without the ability to concentrate oxygen in specialized molecular carriers, the animal depends entirely on oxygen dissolved directly in the plasma. For that to work, the environment needs to supply oxygen at a concentration high enough to saturate the plasma in metabolically adequate quantities. Only the ultra-oxygenated polar waters provide that condition. Moving an icefish to any other ocean on the planet is essentially a death sentence by gradual suffocation. The heart of Kinocephalus aceratus is proportionally the largest among vertebrates, 5 times more voluminous than that of fish of equivalent size that do have hemoglobin. To compensate for the inefficient oxygen transport of plain plasma, the cardiovascular system needs to move far larger volumes of blood with each cardiac cycle. Cardiac output, the amount of blood pumped per minute, is up to 5 times greater than in comparable fish. Blood vessels are correspondingly wider. Capillaries have an increased diameter to reduce flow resistance. The low viscosity of blood without red cells makes that accelerated flow easier. The entire cardiovascular architecture was redesigned to solve through volume and speed the problem that other vertebrates solve through molecular concentration. The absence of red blood cells, erythrocytes, in the blood of adult icefish is complete. Young larvae have vestigial traces of erythrocyte precursors, suggesting that the genes weren't fully deleted, but rather silenced during development. The translucent plasma circulating through the cardiovascular system of these fish is visible to the naked eye. Gills, which in other fish are red from hemoglobin, show white or pale pink coloration in icefish. The heart, vessels, and cardiac musculature, normally reddish from myoglobin, have also lost pigmentation in some species. Kinocephalus aceratus specifically lacks both hemoglobin and myoglobin in the cardiac muscle, making its heart literally white. Myoglobin, the protein that stores oxygen in the skeletal and cardiac muscle of vertebrates, is also absent in the heart of Kinocephalus aceratus, although some other icefish species retain it partially. This double loss is physiologically risky. Cardiac muscle is metabolically intense and depends on a local oxygen reserve for continuous function, especially during high-frequency rhythmic contractions. The solution the animal arrived at appears to involve mitochondria, the organelles that produce energy, at exceptionally high density in cardiac cells, maximizing the efficiency of oxygen utilization from the plasma. More mitochondria per cell means less oxygen needed per unit of work performed. The skin of icefish actively contributes to oxygen absorption. With no scales in several species, or with very reduced scales, and highly vascularized, the skin surface functions as a supplementary respiratory organ, absorbing dissolved oxygen directly from the surrounding cold water. Estimates suggest that up to 40% of the oxygen consumed by Kinocephalus aceratus is absorbed through the skin rather than the gills. This cutaneous mechanism exists in other fish to a lesser degree. In icefish, it's been amplified to the point of becoming a significant component of the total respiratory system. The entire body surface transforms into a gas exchange surface. Antifreeze proteins, AFPs, round out the icefish's adaptations. The freezing point of Antarctic seawater reaches approximately minus 1.9 degrees Celsius. Fish body fluids would freeze at that temperature without additional protection. AFPs bind to nascent ice crystals and inhibit their growth, lowering the effective freezing point of body fluids without altering the melting point, a phenomenon called thermal hysteresis. This molecular solution was discovered independently in Antarctic and Arctic fish, making it a classic case of convergent evolution. Similar environmental pressure producing similar molecular solutions in unrelated lineages. The icefish genome reveals the molecular history of hemoglobin loss. The globin genes, the gene family that includes hemoglobin and myoglobin, were progressively silenced and deleted over approximately 77 million years of isolation in the Antarctic Ocean. During that period, mutations that would have been lethal in any other environment became neutral or even advantageous. Without hemoglobin, red blood cell production was no longer necessary, freeing up energy resources previously dedicated to synthesizing those cells. The loss that seems catastrophic was, in this specific environment, a metabolically economical simplification sustained by the oxygen richness of polar waters. The icefish is a living physiological paradox, a vertebrate that breaks the most fundamental rule of vertebrate biology and survives precisely because the environment in which it breaks that rule is the only one capable of sustaining that violation. Take it out of cold water, and it dies of suffocation. Keep it where it evolved, and it thrives without the molecule that all of animal physiology considers indispensable. This organism demonstrates that indispensable is a contextual category, not an absolute one. What is essential in one environment can become superfluous in another sufficiently different one, and eventually disappear without leaving anything behind but translucent plasma flowing through wide veins in a white heart that beats harder than any red equivalent. The Dumbo octopus, *Grimpeteuthis *SPP, is the deepest living octopus on the planet. A specimen was confirmed at 6,957 meters depth in the Java Trench, establishing the genus's hadal distribution. The name comes from two ear-shaped fins it uses for propulsion with slow, rhythmic beats. Unlike other octopuses, it has no ink sac and doesn't use jet propulsion to move. It has blue blood containing hemocyanin, a copper-based protein more efficient than hemoglobin at transporting oxygen in cold, low-oxygen environments. It swallows prey whole without chewing. Its gelatinous body distributes pressure uniformly, the mechanism that allows it to exist where any rigid structure would be crushed. The hadal zone, depths below 6,000 meters, corresponding mainly to ocean trenches, accounts for less than 1% of the ocean floor, but concentrates some of the most extreme conditions on the planet. Hydrostatic pressure at 7,000 meters equals roughly 700 atmospheres. Every square centimeter bears a force equivalent to the weight of 700 kilograms. Rigid structures with internal cavities collapse, gases compress to minimal fractions of their volume, inadequate cell membranes solidify. Getting to these depths is already a considerable engineering challenge for robotic equipment. Permanently inhabiting these depths requires biological solutions that work around, rather than resist, the physical forces involved. The Dumbo octopus found an elegant solution, become, in large part, pressure itself. The gelatinous body of Grimpeteuthis is the key to its hadal distribution. Biological gelatin, tissue made up mainly of water and flexible structural proteins like collagen, is essentially incompressible and transmits pressure uniformly in all directions without structural deformation. There are no air cavities to collapse, no rigid structures to fracture under asymmetric compression. The animal is mechanically, almost continuous, a mass of organized biological fluid that behaves under extreme pressure much like water itself. This strategy of becoming like the environment, rather than resisting the environment, is a recurring theme among hadal organisms and represents a convergent solution to the same physical problem. The fins that give the animal its name, two lateral expansions of the mantle that resemble the ears of the fictional elephant Dumbo, function as lift and propulsion surfaces. Slow, rhythmic beats of these fins generate thrust with considerable energy efficiency in an environment where food resources are scarce and energy conservation is imperative. Conventional octopuses use jet propulsion, contracting the mantle to forcefully expel water, producing rapid but energetically costly acceleration as their primary escape and attack mechanism. Grimpeteuthis did away with that system. With no known predators at 7,000 meters and no prey requiring high-speed pursuit, explosive speed became unnecessary. Fin-controlled hovering replaced the jet. The absence of an ink sac in Grimpeteuthis reflects the same adaptive logic. Ink is a defense against predators, an opaque cloud that obscures the attacker's field of vision while the octopus escapes. With no relevant predators in the hadal zone, the metabolic investment in producing and storing ink ceased to offer any adaptive return. Structures without function under neutral selective pressure tend to regress. The same principle that eliminated eyes in cave fish and pigmentation in abyssal organisms. The Dumbo octopus lost its ink the same way the ice fish lost hemoglobin. Gradually, over generations, in an environment where the absence didn't matter. Hemocyanin, the respiratory pigment of Grimpeteuthis, differs fundamentally from hemoglobin. Instead of iron, it uses copper as the central oxygen-binding atom. When oxygenated, hemocyanin turns blue. responsible for the characteristic bluish color of octopus, squid, and crustacean blood. Hemocyanin dissolves directly into the plasma rather than being confined to cells, and its oxygen-binding efficiency increases at low temperatures and slightly acidic pH, conditions that prevail in cold, deep waters. For Grimpoteuthis, hemocyanin is a respiratory pigment optimized for the exact environment it inhabits, just as the specialized hemoglobin of Riftia pachyptila is optimized for the hydrothermal environment. The Dumbo octopus feeds by swallowing prey whole, copepods, amphipods, polychaetes, and other small abyssal invertebrates. Without a functional radula to scrape or grind food, the animal catches prey with its tentacles and directs it straight into the esophagus. This whole prey feeding strategy eliminates the need for elaborate chewing apparatus, simplifying oral anatomy, and reducing the metabolic costs of structures that require continuous upkeep. In an environment where finding any prey at all can be an infrequent event, swallowing quickly and completely maximizes the caloric return from each successful catch. Reproduction in Grimpoteuthis has an additional quirk. Females carry eggs at different stages of development simultaneously, suggesting the ability to store sperm and fertilize eggs continuously and asynchronously over time. There's no defined breeding season. Eggs are released individually when local conditions appear favorable, a strategy that spreads reproductive risk rather than concentrating it in periodic events. Each egg is relatively large and yolk-rich, providing the hatchling with enough nutrition to emerge already as a fully functional miniature version of the adult, with no pelagic larval phase. This eliminates the need for larvae to ascend to surface layers, a dangerous journey that would expose young hadal animals to environments completely unlike those they're adapted to. The bathymetric range of the genus Grimpoteuthis, with species documented between 1,000 and nearly 7,000 meters, represents one of the most extensive vertical ranges among cephalopods. Different species occupy distinct depth bands within that interval, suggesting graduated ecological specialization as depth increases. The species confirmed at 6,957 meters in the Java Trench set the depth record for any octopus, surpassing the previous limit by more than 1,000 meters. That record, obtained through low-light cameras mounted on autonomous vehicles, documented the animal moving actively in the hadal zone, demonstrating that extreme depth isn't a physiological limit, but a functional habitat. The Dumbo octopus closes an arc that began with the volcano snail. Each organism presented here inhabits a distinct extreme: volcanic heat, an impossible thermal gradient, steadily increasing abyssal pressure, but all share the same fundamental solution. Not resistance to the environment, but integration with it. The snail incorporates the volcano's iron into its shell. The Pompeii worm distributes the heat across its body rather than blocking it. The Yeti crab cultivates the microbial environment on its own arms. Riftia internalizes the hydrothermal chemistry into an entire organ. The icefish dissolves into the properties of polar water. The Dumbo octopus becomes, mechanically, almost indistinguishable from the pressure that surrounds it. Life in Earth's extreme environments isn't survival against the environment. It's a way of becoming the environment. The swimming sea cucumber, Enypniastes eximia*, spends most of its time swimming in open water, only descending to the sediment to feed quickly. Its body is semi-transparent. The internal organs are visible through the body wall. Breathing happens through the cloaca, the posterior opening. Water enters through the anus, oxygen is extracted by the internal water lungs, and the water is expelled. When threatened, it produces bioluminescence through hundreds of granular bodies inside its gelatinous skin. A broad touch triggers a full-body light response. The skin is fragile. Strong physical contact easily causes it to shed in a glowing cloud that floats in place and distracts the predator while the animal slips away. The defense system requires the animal to lose parts of its own body in order to survive. Picture an organism whose last resort immune system is to come apart. Not a metaphor, an actual physiological process where the entire skin detaches in a luminous cloud while the naked body escapes into the darkness. Enypniast's eximia, a holothurian belonging to the order Elassopodida, inhabits the water column between 500 and 5,000 meters deep and represents one of the most unusual survival strategies among echinoderms. Its translucent body, digestive organs, gonads, and vascular structures visible as silhouettes through the gelatinous body wall, navigates the deep ocean by beating an anterior swimming structure that resembles an inverted crown, feeding on organic sediment deposited on the bottom and returning to the water column between meals. Breathing through the cloaca, a multipurpose posterior opening that simultaneously serves as a digestive outlet and respiratory inlet, is a trait shared by many sea cucumbers, but Enypniastes employs it with particular efficiency. Seawater is actively pumped in by the cloacal musculature, travels through a branching network of tubes called respiratory trees or water lungs, where oxygen is extracted into the coalomic fluid, an internal fluid that serves as the functional equivalent of blood, and then the oxygen-depleted water is expelled. The reversal of respiratory flow, entry through the posterior rather than anterior opening, seems counterintuitive but offers a functional advantage. It keeps the anterior end, used for capturing food, free from being tied up in respiratory functions. The body transparency of Enypniastes deserves separate analysis from its defense system. Pelagic open ocean organisms face the absence of substrate for hiding. No reefs, no sediment, no vegetation to conceal their presence. A recurring strategy in this environment is transparency. Tissues that transmit light rather than absorb it make the organism virtually invisible to predators that detect prey visually. Gelatinous water with a refractive index similar to that of the surrounding seawater minimizes the optical disruption that would signal the presence of a solid body. Inevitably opaque internal organs remain as the only visible trace, small dark structures apparently suspended in nothing, with no discernible body outline connecting them. Bioluminescence in Enypniastes is produced by photophores, specialized cells or granular bodies distributed throughout the skin. These photophores contain luciferin, a chemical that, when it reacts with the enzyme luciferase in the presence of oxygen, releases energy in the form of visible light photons. The emitted spectrum is typically concentrated in the blue-green range, the wavelength that penetrates deepest in water and that the eyes of deep-sea organisms are especially sensitive to detecting. A mechanical touch anywhere on the body triggers a cascade activation of the photophores that spreads across the entire skin within seconds, lighting the animal up completely before the skin begins to detach. The shedding of the skin, a process called tegumentary autotomy, involves the programmed rupture of connections between the outer skin and the underlying muscle layers. This rupture isn't accidental. It occurs through an active mechanism that selectively weakens the tissue interface in response to the mechanical stimulus of predatory contact. The detached skin continues to emit bioluminescence for a period after separation, behavior that maximizes the distraction effect, keeping the predator focused on a glowing cloud that doesn't correspond to any nutritious organism, while the sea cucumber's bare body retreats into the darkness. The sacrificed skin is expendable. The animal regenerates a complete new skin over the course of days to weeks. The regeneration that follows tegumentary autotomy is made possible by the general biology of echinoderms, the phylum that includes starfish, sea urchins, and sea cucumbers, which features exceptionally well-developed regenerative capacity among animals. Starfish regenerate entire arms. Some sea cucumbers expel their internal organs as a defense and rebuild them within weeks. Enypniast's eximia regenerates its skin with no apparent long-term compromise to the animal's integrity. This regenerative potential is a necessary condition for tegumentary autotomy to be a viable defensive strategy. Without efficient regeneration, losing the skin would be a prohibitive cost rather than a survival investment. Enypniast's diet consists of organic matter deposited in the sediment, particles from dead organisms, feces from pelagic animals, and terrigenous material that continuously sinks from the surface. This material, collectively known as marine snow, is the main vector for transferring energy from the sunlit photic zone to the deep ocean. The sea cucumber periodically descends to the sediment, passes its oral apparatus over the bottom collecting organic particles, and returns to the water column. Studies with deep-sea cameras have documented this behavior. The animal literally grazes on the sediment for brief periods before ascending, alternating between the relative safety of the water column and the food productivity of the bottom. The bioluminescent granular bodies of enypniasts have been studied as a model for understanding bioluminescence in deep-sea organisms. Unlike squid and fish that have complex photogenic organs with lenses, reflectors, and filters, enypniasts' system is relatively simple. Granules dispersed throughout the skin that emit diffuse light rather than a directed beam. This apparent structural simplicity produces a dramatic visual effect precisely because the entire skin becomes the emitter, creating full-body illumination rather than localized bright spots. For a predator attacking in the dark, the sudden appearance of an entirely luminous body produces enough of a startle response for the separation and escape to take place. Enypniast's eximia represents the dynamic counterpoint to the volcano snail that opened this series. Where the snail built impenetrable iron armor, the sea cucumber built disposable, luminous skin. Where the snail incorporates the environment as permanent protection, Enypniast sacrifices part of itself as temporary protection. Two opposite solutions to the same fundamental problem: surviving in an environment where predation is a constant threat. The fact that organisms so different arrived at strategies so contrary to each other demonstrates the breadth of solutions biology accommodates when the only criterion is to work well enough to reproduce before being eaten. The hydrothermal vent mussels, *Bathymodylis *SPP, solve the problem of food scarcity differently from tube worms. Instead of housing bacteria exclusively inside themselves, they maintain bacteria in their gills that use both hydrogen sulfide and methane as energy sources. Dual symbioses involving the simultaneous presence of sulfur-oxidizing and methane-oxidizing bacteria within the same host cells are a widespread feature of Bathymodilin mussels from hydrothermal vents and cold seeps in the Atlantic Ocean. They are the only known bivalves with this dual chemosynthetic symbiosis. The mussel's symbionts are acquired anew each generation through infection by free-living forms. The larvae need to find another active hydrothermal vent on an ocean floor where these vents are rare and far apart before starving to death. Bivalves, mollusks with shells divided into two articulated valves, like oysters, clams, and common mussels, generally feed by filtration. Water passes through the gills, organic particles get trapped in mucus and are transported to the mouth. This system works perfectly where there is phytoplankton and organic matter in suspension. At 2,500 meters deep, 0 kilometers from any sunlit bottom, and hundreds of kilometers from continental coasts, the system collapses for lack of raw material. The genus Bathymodilus solved this impasse by transforming its own gills, originally filter-feeding structures, into biochemical reactors inhabited by bacteria that produce food from inorganic compounds. The gill stopped being a sieve and became a farm. The dual symbiosis is the most extraordinary trait of Bathymodilus. Two functionally distinct groups of bacteria coexist within the same gill cells of the mussel. sulfur-oxidizing bacteria, which metabolize hydrogen sulfide, and methane-oxidizing bacteria, which metabolize CH4. Each group uses a different substrate and produces organic biomass that is transferred to the host. This functional redundancy confers considerable metabolic flexibility. When a hydrothermal vent supplies more sulfide than methane, the sulfur bacteria dominate. When the composition of the fluids changes, the methanotrophs increase their contribution. The muscle doesn't depend on a single energy source. It has two, operating in parallel within the same tissue. Host cells simultaneously housing two distinct types of symbionts face a considerable immunological challenge. Animal immune systems generally recognize and attack bacteria as invaders. Symbiotic organisms need to be tolerated by the host while genuine pathogens are eliminated. In Bathymodialis, this distinction is maintained through molecular mechanisms that are still not fully understood, possibly involving local suppression of the immune response in gill cells, molecular signals from the bacteria that mimic the host's self-markers, or a combination of both. Simultaneously maintaining two distinct bacterial types without immunological confusion represents regulatory sophistication that invertebrate immunology researchers continue to investigate. The horizontal acquisition of symbionts, rather than direct maternal transmission, imposes a dramatic cost on the Bathymodialis life cycle. Larvae hatch without symbiotic bacteria. They need to find an active hydrothermal environment where free-living chemosynthetic bacteria are available for colonization before exhausting their yolk reserves. Hydrothermal vents are sparsely distributed along mid-ocean ridges, underwater mountain ranges that run along the ocean floor for tens of thousands of kilometers, but with active vents separated by distances that can reach hundreds of kilometers. The larva, tiny and with limited mobility, must cover enough distance to locate an active vent before its starvation window runs out. Deep ocean currents are the dispersal mechanism available to Bathymodialis larvae. Circulation patterns along mid-ocean ridges can transport larvae between hydrothermal vents on timescales compatible with their survival window, but the reliability of this transport varies with local geology and tectonic activity. Some Bathymodialis populations show genetic connectivity between hydrothermal vents separated by hundreds of kilometers, confirming successful larval dispersal over considerable distances. Other populations show genetic isolation, suggesting that oceanographic or geological barriers regularly interrupt larval flow. For every larva that finds an active vent, an unknown number, likely far greater, perish without ever establishing symbiosis. Cold seeps, locations where methane and hydrogen sulfide-rich fluids emerge from the ocean floor without the heat associated with black smokers, host Bathymodialis communities distinct from hydrothermal vent communities. In these environments, water temperature stays close to that of the surrounding deep ocean, but the chemistry of the fluids provides the substrates needed for chemosynthesis. Cold seep-specific Bathymodialis species show different proportions between the two symbiotic groups compared to hydrothermal species, a reflection of the different chemical composition of the available fluids. The same dual symbiotic platform adapts to different substrate sources by adjusting the relative abundance of each bacterial group. The Bathymodialis shell has a composition and microstructure comparable to those of surface mussels, calcium carbonate in a crystalline arrangement of calcite and aragonite. Growth, however, is considerably slower than in shallow water equivalents, where higher temperature and abundant food supply accelerate mineral deposition. At hydrothermal vents, temperature can be locally elevated, slightly speeding up metabolism, but the energy available for somatic growth competes with the demands of maintaining the bacterial symbiosis. Analysis of growth rings in the shell makes it possible to estimate the age and growth rate of individuals. A method similar to the use of tree rings in dendrochronology applied to abyssal conchology. Bathymodylis populations around the mid-Atlantic black smokers, particularly at the Lucky Strike and Rainbow hydrothermal fields, have been extensively studied by European expeditions over decades. These populations revealed that the mussel is not merely a passive consumer of bacterial production. It acts as an ecosystem engineer, forming dense aggregations that modify hydrothermal fluid flow, create microhabitats for other organisms, and accumulate biomass that sustains predators like crabs and octopuses. The experimental removal of Bathymodylis aggregations alters the structure of the entire local community, evidence that this bivalve's ecological role extends well beyond its own nutrition. From the volcano snail's iron armor to Bathymodylis' dual bacterial farm, each organism in this sequence has redefined a category that seemed fixed. Digestion without a gut, a vertebrate without hemoglobin, skin as a defensive sacrifice, a gill as a dual fermentation reactor. The pattern that emerges is not one of curious exceptions to normal biology. It's an entirely alternative biology, developed in the cracks of the planet where the rules that describe life on the surface simply don't hold up. While the sunlit surface runs on photosynthesis and oxygen, this other underwater world runs on pure chemistry, extreme pressure, and symbioses that redefine where the organism ends and the environment sustaining it begins. The Greenland shark, Somniosus microcephalus*, takes up to 150 years to reach sexual maturity, longer than any other known vertebrate. Most captured adults have a parasite, Omidocoida elongata*, attached to the cornea of one or both eyes, causing permanent vision damage. The parasite feeds on ocular tissues. The shark, with minimal metabolism and moving at less than 3 kilometers per hour, tolerates the infection without any significant immune response. The flesh contains high concentrations of trimethylamine oxide, making it neurotoxic if consumed fresh. Inuit peoples ferment the meat for months before eating it, in a process called hakarl. The oldest dated specimen was estimated at 512 years old, possibly born before Leonardo da Vinci. Sexual maturity at 150 years means that a Greenland shark born today won't reproduce until 2175. No human alive will witness this animal's first mating. This timescale puts Somniosus microcephalus in a category of its own among vertebrates. Bowhead whales, which also reach exceptional longevity, reach sexual maturity between 20 and 25 years. Giant tortoises reproduce after 3 to 5 decades. The Greenland shark multiplies those figures by 5. The root cause is an extremely reduced metabolism, a rate of biochemical processes so slow that growth, development, and the accumulation of cellular damage occur on a timescale that defies comparison with any other known vertebrate. Temperature is the primary regulator of metabolism in ectothermic organisms. Animals that don't generate body heat internally and whose biochemical processes speed up or slow down with ambient temperature. The Greenland shark inhabits Arctic and subarctic waters between -1 degree Celsius and 10 degrees Celsius, spending most of its time near the lower end of that range. At these temperatures, enzymatic reactions occur at a fraction of the speed they would in temperate waters. Somatic growth, cell division, protein synthesis, all fundamentally temperature-dependent processes, progress at a correspondingly slow pace. The animal doesn't choose to grow slowly. The physics of temperature impose that rhythm on the entire metabolism. Dating the Greenland shark used carbon-14 incorporated into the lens of the eye. The lens is metabolically inert tissue after formation. Proteins deposited during the embryonic phase remain without renewal throughout the animal's entire life. Atmospheric carbon isotopes have varied over the decades, with specific markers corresponding to periods of atmospheric nuclear testing in the 1950s and 1960s. By comparing the proportion of carbon-14 in the lens against reference curves calibrated by these historical markers, researchers estimated the ages of specimens with enough precision to identify the oldest individual in the sample as approximately 392 years old, with a confidence interval that extends the upper limit to 512 years. Born, possibly, in the early 16th century, the parasite Amatocoida elongata is a copepod, a tiny crustacean related to crabs and lobsters, but adapted to ocular parasitism. Adult females attach themselves permanently to the shark's cornea through an anchoring structure that penetrates the ocular tissue. The parasite's body, elongated and whitish, hangs freely in front of the host's eye as the animal moves. Feeding occurs through direct absorption of corneal fluids and tissues. The resulting damage is progressive and permanent. Corneal opacity develops at the attachment site, compromising light transmission. In bilateral infestations, present in a significant proportion of captured adults, visual impairment is severe in both eyes simultaneously. The Greenland shark's immunological tolerance of Amatocoida elongata infestation remains partially unexplained. Vertebrate immune systems generally respond to foreign tissue in prolonged contact with an inflammatory reaction that, while sometimes destructive, tends to isolate or eliminate the invading agent. In Somniosus microcephalus, this response appears suppressed or absent around the parasite's attachment site. Hypotheses include local immunosuppression mediated by the parasite itself, a mechanism documented in other parasites that manipulate the host's immune response to ensure their own survival, or simply a constitutively slow immune response, consistent with the animal's generally reduced metabolism. A swimming speed below 3 kilometers per hour, roughly half the pace of a comfortable human walk, doesn't seem sufficient to catch agile prey. Yet stomach content analysis has revealed fish, seals, reindeer, and even polar bears among the Greenland shark's food items. The most likely explanation involves predation of sleeping or dead animals, ambush feeding in currents that concentrate prey, and possibly the capture of already weakened animals. Carcasses that sink also constitute a relevant food source. The slowness doesn't prevent feeding, it redefines the strategy. Rather than active pursuit, the Greenland shark operates as a highly energy-efficient opportunistic scavenger, spending the minimum to get the maximum. Trimethylamine oxide, TMAO, accumulates in the tissues of Somniosus microcephalus at concentrations that make the flesh neurotoxic if consumed fresh. This compound functions as an osmoregulator in elasmobranchs, sharks and rays, compensating for the osmotic difference between body fluids and salt water. In cold water, deep-sea species, TMAO concentrations are particularly high because the compound also stabilizes proteins against denaturation by hydrostatic pressure. When metabolized by an organism that consumes the flesh, TMAO converts to trimethylamine, which affects the central nervous system, causing symptoms similar to severe intoxication. Dogs fed fresh Greenland shark meat show pronounced motor incoordination. Hakarl, fermented Greenland shark meat consumed in Iceland, results from a detoxification process developed by Nordic communities over centuries. The meat is buried in sand or gravel for weeks to months, allowing body fluids rich in TMAO to drain and bacteria to begin fermentation that breaks down the toxic compound. It is then hung outdoors for additional months to cure. The final product has an intense characteristic ammonia smell, a byproduct of bacterial degradation of nitrogen-containing compounds, and a flavor that visitors frequently describe as extreme. The toxicity is eliminated, the protein remains usable. The empirical knowledge of Arctic peoples figured out a processing method that modern chemistry would only explain decades later. The Greenland shark closes this list with a temporal proposition that reshuffles all the others. The volcano snail exists at 350 degrees Celsius. The Pompeii worm splits 78 degrees Celsius across its own body. The Dumbo octopus inhabits 700 atmospheres. All of those extremes are spatial, conditions of the environment the organism lives in. Somniosus microcephalus adds a temporal extreme. 512 years of continuous existence, partially blind, moving at a slow walking pace, carrying parasites on its eyes without any significant response, accumulating half a millennium of silent biological history in the dark waters of the Arctic. No laboratory replicates that scale. No experiment observes it from start to finish. The Greenland shark is the only organism in this series that existed before the science describing it existed. The volcano snail builds iron armor next to 350 degrees Celsius chimneys. The Pompeii worm endures a 78 degrees Celsius gradient across its own body. The Yeti crab farms bacteria on its arms as a food supply. The giant tube worm is 3 meters long with no digestive organs. The icefish has colorless blood and a heart 5 times larger than that of comparable fish. The Dumbo octopus floats at 7,000 meters on ear-shaped fins with blue blood. The sea cucumber breathes through its anus and sheds its own skin when touched. The vent mussel maintains 2 simultaneous bacterial symbioses to survive without sunlight. And the Greenland shark spends 150 years growing until it can reproduce, carrying parasites on its eyes while slowly swimming in polar darkness. The entire known range of the volcano snail is equivalent to 2 American football fields. And this animal with its magnetic shell represents just one point on a list of organisms that made biologically possible what should be impossible. 9 organisms. 9 radically different solutions to the same fundamental problem, existing where conditions should make existence impossible. None of them experience the environment they live in as a challenge. For each one, the sulfide chimneys, the polar ice, the abyssal darkness, and the hadal pressure are simply the world. The strangeness is the surface observer's perspective, not the condition of the organism being observed. What this list reveals, accumulated organism by organism, is not a collection of biological curiosities. It's systematic evidence that the limits biology has drawn for itself are provisional limits, open to revision with every expedition that goes down deep enough. Consider what was dismissed as impossible before each discovery. That an animal could build a mineralized structure with iron sulfide, impossible until the volcano snail. That a metazoan could tolerate a 78 degrees Celsius gradient along its own body, impossible until the Pompeii worm. That an adult vertebrate could operate without hemoglobin, impossible until the Antarctic icefish. That a 3-meter-long invertebrate could entirely lack a digestive organ, impossible until Riftia pachyptila. The word impossible, in the context of extreme environment biology, has proven to be almost invariably provisional, a declaration of temporary ignorance about what exists outside the immediate field of view. Symbioses represent the thread running silently through the entire list. The volcano snail houses bacteria in its esophagus that supply the minerals for its armor. Riftia pachyptila replaced its entire gut with an organ packed with bacteria. The Yeti crab farms bacteria externally on its arms. Bathymodialis maintains 2 bacterial species simultaneously in its gills. The Pompeii worm carries a bacterial coat of still debated function. In practically all of these organisms, the boundary between the animal and its associated microbes is functionally porous. Host metabolism and bacterial metabolism are intertwined to the point where deciding where one ends and the other begins is arbitrary. Pressure, temperature, chemical toxicity, and scarcity of sunlight are the 4 variables that define deep extreme environments. And each organism on this list faces distinct combinations of these variables with solutions that don't repeat themselves. There's no universal solution to the extreme. The gelatinous body that works for the Dumbo octopus at 700 atmospheres would be useless for the ice fish that needs oxygenated plasma in polar water. The volcano snail's iron sulfide armor would be metabolically unviable for the Greenland shark that minimizes energy expenditure to the maximum. The diversity of solutions reflects the diversity of selective pressures. Each niche shapes its occupant in an unrepeatable way. Biotechnology is watching these organisms with growing interest and precise practical reasons. Heat shock proteins from Alvinella pompigana are of interest to the pharmaceutical industry because they stabilize biological molecules at temperatures that would destroy conventional equivalents. The three-layer shell architecture of the volcano snail is inspiring composite material designs for armor and impact absorption. The extracellular hemoglobins of Riftia pachyptila, capable of simultaneously transporting oxygen and sulfide without mutual interference, suggest molecular architectures for artificial oxygen carriers. The Dumbo octopus's hemocyanin, efficient at low temperatures and high pressures, has implications for bioprocessing under extreme conditions. Abyssal biology is not merely a curiosity. It's a repository of molecular engineering tested by rigorous selection over geological time. The 512-year-old Greenland shark and the volcano snail restricted to 2 football fields of habitat represent opposite extremes of the same condition. Rarity. Temporal rarity, half a millennium of individual life, and spatial rarity, a geographic range that fits in an area smaller than most city neighborhoods. Both forms of rarity make these organisms extraordinarily vulnerable. A shark that takes 150 years to reproduce for the first time doesn't bounce back quickly after a disruption. A snail whose entire habitat equals 2 sports fields can be wiped out by a single deep-sea mining operation. Getting to know these organisms and losing their existence before fully understanding them would be events that could occur on the same human timescale. Astrophysics and extreme environment biology converge on one question. If life arose on Earth under conditions as diverse as 350 degrees Celsius black smokers and -1 degree Celsius polar oceans, what conditions are necessary for life to arise on other worlds? The answer these 9 organisms collectively suggest is unsettling in its breadth: very few. Liquid water, a chemical gradient, and sufficient time appear to be the minimum requirements. Europa, Jupiter's moon with a subsurface ocean heated by tidal forces. Enceladus, Saturn's moon with vapor plumes ejecting organic compounds into space. Both satisfy these minimum criteria. The biology we've found at the bottom of Earth's oceans has become the most solid argument available for searching for life on those worlds. What connects the volcano snail to the 512-year-old shark, passing through the worm without a gut, the fish without hemoglobin, and the octopus at 7,000 meters, is more subtle than the list of records each one holds. It's the repeated demonstration that biological systems find solutions where physics seems to leave no room for them. Each of these organisms inhabits the intersection between what thermodynamics allows and what selection shapes over geological time. They aren't miracles, they're optimizations. Local solutions to local problems, arrived at by the accumulation of variation over generations counted in the millions. The impression of impossibility they provoke results from comparing the human scale of time and perception with the scale at which biology operates. The narrative that began with an iron shell at a volcanic chimney 2,900 meters down returns, inevitably, to the same starting point. Not because it's circular by design, but because the organisms that compose it collectively point to the same conclusion. The definition of habitable is always smaller than the set of places where life actually lives. Every expedition that descends below the limit where sunlight reaches finds organisms that shouldn't exist according to the criteria in place up until that discovery. The list presented here isn't exhaustive. It's only what has been found so far, in the small fraction of the ocean floor that human instruments have already inspected with sufficient resolution. What remains to be discovered, in the unvisited fissures and unmapped trenches, will likely revise once more the limits this list already revised. And the volcano snail, unmoved in its iron armor beside the chimney that forged it, will keep on existing where it shouldn't, as it always has.